This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 29 for April 24th, 2009. From New York City, I'm Vincent Racaniello.
I'm Dick Despommier.
And from Western Massachusetts, I'm Alan Dove.
And it's gorgeous out.
It is beautiful.
It's beautiful here, too.
It's the first great day of the year.
But can you tell me, gentlemen, this week it's been 10 degrees, right?
Yeah.
What is it going to be this weekend?
80.
Well, you're changing measurements, Dick.
It'll be close to 30.
Why do we have such massive jumps? Is this global warming?
No, we've had this fluctuation in April for the last 20 years.
It's typical.
It is.
Yeah, it's normal for the fluctuation to occur. What we have had from global warming is that last frost or first frost has shifted. The growing season has gotten longer.
So this is normal temperature fluctuation at this time.
It is. Yeah. It is. But what is abnormal is that every year, and I think this year was no exception, the cherry blossoms in Washington have bloomed earlier than the year before.
And all the blossoms.
And the last year was the hottest year on record compared to the year before that, and the year before that was the hottest year on record compared to the year before that, et cetera, et cetera. So that's a trend.
Anyway, it's really nice out.
Yeah, it's gorgeous.
All right, we have a couple of news stories, and then we have a bunch of reader email. We probably won't get through it all, but I'm thinking once a month we should do a Q&A, should we call it? Which is what today is.
Absolutely.
All right, the first story, Alan, you picked up. Discovery of insect and human dengue host factors.
Huh.
Yes. So this just came out in Nature, and these folks did a sort of a shotgun approach with RNAi. So they're using inhibitory short sequences of RNA to shut off the genes in a cell, or not shut off, but knock down is the term.
Dampen.
You substantially reduce the expression of the gene by using these little short interfering pieces of RNA that bind to the transcript and muck it up. So the approach they use, this is working with dengue virus. One of the questions is, well, what does it need in a host cell in order to replicate? What are the host factors that are required for this virus to make more of itself? There hasn't been a whole lot of work on this because it's kind of hard to look at. You've got a virus that can replicate in human cells for its life cycle, or it can replicate in insect cells because it's mosquito spread. They use Drosophila melanogaster cells, fruit fly cells, which fruit flies are not really very well known as a vector of dengue fever virus. They are insect cells.
They're a permissive cell line.
They're a permissive cell line, and of course the genetics of Drosophila has been studied for a century or so, whereas the genetics of mosquitoes is a little harder to work with. So they use this Drosophila cell line and progressively knocked out 22,632 different RNAis that they used in this thing. So they're doing a real...
Good heavens.
Panel type of approach, knocking down all the possible individual genes.
Why not just pour Clorox on it?
Well, they wanted to knock down one at a time.
One at a time, not the whole cell.
And they did 22,000 some odd different experiments?
One at a time. Yeah, they did. Probably in duplicate, too. Dick.
That's amazing.
Yeah, it's a lot. But it's mechanized in 96 well plates or even greater. So you can do this. And you look for in each one if the virus grows, right?
Yeah,
right.
Exactly.
I want to do this, too, with our viruses.
Yeah.
Expensive.
So tell us, what did they find?
Well, they found a whole bunch of candidate dengue virus host factors, or DVHFs, as they're called. These are genes that appear to be necessary. You knock one of them down, and you don't get dengue replication, or you get very little dengue replication in the cells. And what I thought was interesting was they looked at this panel of them, and they found that 82 of these had recognizable human homologs. Then they did another RNAi screen in human cells, and they found 42 genes of that set that are human dengue virus host factors, or apparent human dengue virus host factors. So they now have this whole set of host genes that are probably or possibly involved in dengue virus replication, and a whole bunch of them are new. We didn't know that these genes might be involved, and now it's a whole new set of directions to look for things that you could potentially inhibit to prevent the virus from replicating.
How interesting. Tell me something. Why haven't we applied this logical system of exploration of interference to bacterial infections?
What would you like to do?
There was a sudden silence at the other end.
What do you want to look at is the question.
I'm thinking about bacterial resistance to antibiotics. This might be a new approach.
Well, bacteria don't have the machinery to use RNAi.
Right.
You have to use it in other kinds of cells, plants, insect, mammalian cells.
You can't interfere with bacterial RNA synthesis?
Not by RNAi, no.
Okay.
There are other ways to do that,
right.
yeah, but not by RNAi.
But I meant, if not that, then some other similar mechanism.
Some kind of genome-wide. It may be done. I don't know. This is This Week in Virology, Dick.
I understand
if you
that,
want
but I'm trying to be more of a generalist here.
This Week in Bacteriology is the next term.
We'll make another podcast. If anyone knows the answer,
sorry about that.
Let us know. Dengue is a flavivirus.
It is.
RNA virus. Enveloped causes dengue hemorrhagic fever. It's a big problem, right?
That's
there.
right.
Yeah.
We did a show on that, actually.
We did. A long time ago.
It seems like.
In the old days.
So
this is cool because some of these proteins might be targets for antivirals, but some of them could be interesting to work on in the lab and understand how the virus works.
Yeah. So, Alan, were there any homologs between the Drosophila panel and the human panel? How many overlaps were there?
There were 82. They found these 116 candidate genes in the fly, and they found 82 that had homologs in humans.
And how close were those homologs?
And then they did, they found 42 of those 82 appear to be dengue virus host factors in humans.
Okay. Right. So they knocked those down in mammalian cells.
Right, so they knocked down the 82 in mammalian cells that looked like homologs. Yeah. They found 42 out of that set.
Fascinating.
It's a cool study. Now, a similar thing has been done with West Nile. That was done last year at Yale, actually. This is interesting because insect cells take up RNA very readily. So this library of interfering RNAs is actually a double-stranded RNA library. And you just throw it on the cells. You don't have to do anything fancy.
Got it.
And they will take it up. Whereas mammalian cells, you have to coax to take up RNA. So that's one reason they probably did it in Drosophila cells.
And maybe bacteria don't take it up at all.
And if they did, they wouldn't do anything with it except chew it up.
Exactly.
All right. So the one objection I have is the use of the word factor.
Yes.
Now, this is a holdover from the old days when you didn't know if it was a protein or an RNA or something else, a chemical of some sort not related in a cell that you were looking at, host factors. But now these are proteins because these RNAs target proteins. So we should call them host proteins, right? But virologists, like many other scientists, get locked into terminology. And so host factor, I think it's a lousy word. Okay, I'm going to step down off my soapbox.
This has been the Racaniello factor.
That's right.
Sorry, what the hell is a factor?
The X factor.
Guys, have you immunized your horses against West Nile and equine encephalitis virus?
Lately?
Yeah.
No, all of my polo ponies are in Venezuela.
You don't have to worry. So you give them Venezuelan equine.
Exactly.
Alan, have you immunized your horses?
Well, I've been meaning to get around to it, but it's so tough to schedule these things.
There's an article, let's see, I have to read this to you. The Virginia Department of Agriculture urges all horse owners to vaccinate their animals now against West Nile and Eastern equine encephalitis if they have not done so. How many people have horses? I guess a lot.
Virginia, a tremendous
amount. So the horses can get very sick from these two viruses.
Extremely sick. I think West Nile has a 40% mortality rate in horses.
So there are vaccines for both of these. One is called the Prevenile, Prevenile. Great name.
Right.
And interesting, I looked it up. This is an inactivated vaccine. It's simply propagated in cell culture. It's inactivated with formalin, and then it's injected into the horse. And there's a similar one for EEEV as well. It's interesting. In the recommendations, they say you need to re-immunize the horses every 12 months.
I wonder why.
What do you think of that, Dick and Alan? Why do you think that's so?
I think because they want to sell more vaccine. If it was a live vaccine, attenuated, then it would stay a long time in the host. But apparently this is cleared rapidly and induces very low levels of immunity.
What do you think, Alan?
Well, actually, I was just reading the Prevenile information. I didn't realize it was a killed vaccine, but that might help explain it. They're talking about how it's a chimeric vaccine of yellow fever in West Nile.
Mm-hmm.
And I was working on the assumption that it was attenuated, but if it is killed, then you may not get as strong a response. So it only, but interestingly, in the ad copy, they say it stimulates humoral and cell-mediated response for strong, long-lasting protection with a single dose.
Yeah, long-lasting is a year, basically.
It's a relative term, yes.
Well, I guess it's not bad, but it is a killed vaccine because it's very easy to make a killed vaccine. To attenuate it, they would have to do a lot of research. And
Right.
This was easier. But I think it's 12 months because probably it's not very immunogenic.
Right.
It hasn't been developed all that much. If this were being developed for humans, it would probably be researched more extensively. And so the immunity wanes. The first batches of IPV polio vaccine in the 50s only lasted a few years because they weren't optimal. So they could do it better. Or if you went back 4 times with boosts to the horses, it might last longer.
Right.
But they want to make it simple, once a year, so I guess it's not bad. But from a virological viewpoint, it's interesting to consider why you would have to do it once a year. I think that's the first animal vaccine we've talked about. Maybe not rabies, right? We talked about that.
We did talk about rabies.
Dick, did you know there's some swine flu in California and Texas?
I did, and it's a mixture of 3 different animal strains of virus.
Right.
So last week, the 17th, there were 2 cases in California. And then yesterday, I was just about to leave, and I got a Wall Street Journal article, and there were 5 more in Texas. And then I got an email from someone to TWiV asking us to talk about it today. Things move quickly.
Yeah, they saw it on Yahoo News.
There is also, it's not clear whether this is related, but there's a major outbreak of swine flu in Mexico City right now.
Right.
In humans?
In humans.
Well, that's
yes,
interesting.
They've had 40 or 50 deaths from it, and
wow.
they estimate something like 800 infected or something like that.
Well, that must have been going on for some time now, right?
Probably. They just, I guess just yesterday or the day before, they started making a big push they canceled school in Mexico City, which,
Wow.
This is the world's 2nd largest metropolis. It's not a trivial matter that they canceled school.
Exactly.
When did they identify it as swine virus, do you know?
That I don't recall. I had bookmarked the news story, and now I don't have it handy.
Well, when you find it, put it in the our agenda so I can stick it in the show notes if you have a link. I didn't know the Mexico City one.
Yeah.
Well, the American cases, these are swine viruses, and they're unusual because they have mixtures of genes from American pigs and European pigs, which in this particular combination hasn't been seen before. So it's basically a new virus. And all these U.S. infections have been pretty mild, but it sounds like if the Mexican ones are the same viruses, then it's been more serious there.
Yeah.
So I thought it was nothing really based on 5 cases in the U.S. We're doing a lot of intensive surveillance for flu now, more than we ever did. So I figured we're going to pick up strains that we didn't see before. Now, it's interesting to note that none of these people had contact with pigs.
Right.
So at some point, someone did to get this.
Right.
But this shows that it can go person to person. Now, swine flu, I don't know if you guys know this, but swine flu is thought to have begun in 1918-19. It went into pigs from that human pandemic. So the 1918 H1N1 strain went from humans into pigs, and it stayed in pigs, and it's been evolving in pigs for many years, and it goes back and forth. Quite interesting.
Yeah.
We used to think that pigs were the mixing vessels because they're susceptible to both avian and human influenza virus strains. But now, in recent years, there's clear evidence that avian strains can go right into people, so it's not clear if you need pigs anymore. But, for example, the H3N2 strains from 1968 that emerged in humans, they went into pigs in the 90s and have been circulating in pigs. So they're very susceptible. They have receptors for all the flu strains in their respiratory tracts, avian and human.
And I have some breaking news here. It looks like the BBC just updated its story on this. A WHO spokesman said 12 out of 18 samples taken from the Mexican victims showed they died from a virus with the same genetic structure as the one found in the U.S.
Oh. That's interesting.
They do be linked.
And that's serious if you have some deaths there.
Yeah.
Well, I thought this was not an issue because this is the end of the flu season, at least in the U.S., right?
Right.
And, it's starting to get warmer and more humid, and those conditions do not favor transmission of influenza viruses. So I figured even if this virus were starting to come in, maybe it would fizzle out. But Mexico City is a little more south, right, Dick? And
it is a lot more
summer has already begun there.
Yes.
Correct?
Yes. But people live very densely in Mexico City, very densely.
Well, obviously we're going to have to follow this, but that's pretty neat.
Mm-hmm.
Yeah.
I thought I sort of wrote this off, but maybe there's more to it. It just goes to show you can't predict when it comes to viruses. All right, we'll put those links in the show notes. Now, before we go on, a short note from Citrix, who make Go to My PC. I hear this all the time. People think they're chained to their office computer because they have to come in to work every day, get stuck in traffic because you got to reach your computer and do work.
You do.
They don't have time to do the things they want to do.
Nope.
I think it would be nice if people could get to their computer wherever they were, right?
That sounds good to me.
Maybe everyone could stay home a day a week.
I think that would be great.
And of course, Alan gets to stay home 7 days a week.
Alan, do you get any work done at home at all?
Actually, I get all the... Let me put it this way. All the work that I get done happens at home.
Alan is a very structured guy. He doesn't take any distractions. And he obviously is making a living, so I guess he gets work done.
I think he's the envy of both of us.
But for everyone else in the world...
That's right.
Well, there are many journalists, I understand, who work at home. But for Dick and I and others who have to drive into work, many times I'd love to stay home, but the files are on my computer here.
So is there a solution to that?
There is a solution. You can be productive wherever you are. Log on to gotomypc.com, and your office desktop will instantly appear. A couple of clicks of the mouse, you can see your desktop in your office. You can get your email. You could read files. You can even access your work network from home.
Which could be handy even for people like me when we travel.
It could be.
Absolutely.
Except for one little thing, of course.
What's that?
You have to leave your computer on.
You do have to leave it on, and that's not a green thing, right?
It's too bad that we can't turn it on remotely.
Yeah, there probably are ways to do that.
Sure.
I am not criticizing the producer of this product. I think it's a great product. The only caveat I would have is that you have to leave your computer on.
You do have to
you pick up the phone, you call somebody who's there, and you say, hey, can you read
That's a
that might work.
Yeah, but anyway, when we get over the turning on business, we can use GoToMyPC.
Right. Yep.
You can try GoToMyPC free for 30 days, but you must visit gotomypc.com/podcast. That's gotomypc.com/podcast. Free 30-day trial. Check it out. Let us know what you think.
Sounds good.
Polio has spread from Nigeria to 15 African countries.
I saw that.
We're trying to eradicate polio, but there are lots of obstacles. And a number of years ago, Nigeria decided they didn't want to be immunized with the vaccine, so they stopped for a year. Wild polio came back. They started immunizing again, but I fear the damage is done. And now it is spreading from Nigeria to 15 African countries from the end of 2008 to March of 2009. It's spreading into a lot of countries where they had gotten rid of polio. This is wild type 1 and type 3 strains. And there are also, there's a very nice CDC report showing the patterns of spread. They can track where these viruses are going by sequencing them. They have a really good polio laboratory network now as a result of this eradication effort. You can see exactly how they go from Nigeria to all the other countries. And there's actually some importation from India to Africa as well. There's one incident of that. Let's see here what the numbers are. Wild polio types 1 and 3. 32 wild polio importations into 15 African countries, resulting in 96 cases from January to March. Polio transmission in 5 previously polio-free African countries. So that's bad. Now remember, if you have, say, Dick, if you have 100 polio cases, how many people are actually infected?
Oh, hundreds, maybe even thousands.
Yeah, 1,000. At least. So there's a lot of virus circulating.
Same's true for West Nile, by the way.
Yeah, there are a lot of inapparent infections.
Yep.
So this is bad, and it just goes to show that it's really hard to eradicate. The vaccine is good, it works, but you have other reasons, and we've talked about that before.
No question.
So the new WHO motto will be proudly eradicating polio for more than 20 years.
So 1988. I was thinking of the 25th anniversary of HIV was the other day, right, Alan?
Right.
So now 20 years of polio. I don't know. These countries are difficult with polio. I don't know what. I mean, the count is less than 2,000 a year, but still it keeps going. And, we're working very hard to keep everybody immunized, and it's hard to do that in countries where there's no polio. Exactly. So these countries that get importations from Nigeria, the reason is that they reduced the immunization.
Sure.
If they were immunizing everyone, they wouldn't get polio.
Right.
But they say, we don't have polio. So I'm not going to go get immunized.
It's a difficult thing to keep going.
It is. Okay, last time, you remember, we talked about a dead Chinese woman on a Russian train?
We did.
Good name for a book. Huh?
Yes.
Agatha Christie.
Eric had gotten this email while we were talking. It might have been SARS. But it turns out she didn't have SARS. She didn't have flu. She had bacterial pneumonia. Isn't it interesting how nowadays there's always fear of SARS, flu, immediately?
We hear hoofbeats and we immediately look for zebras.
It's very quick. In the old days, well, of course, communication was different, but more science awareness, don't you think?
Yeah.
And particularly virus awareness.
That's right.
People are like that Wall Street Journal article popped right out. They had a phone call with someone at CDC and boom.
Yeah.
In the old days, it seems to me that it would have taken longer to develop a story. Is that right, Alan?
I think it would have. Even the old days of 10 years ago, I think, with the rise of internet news and the 24-hour news cycle and all that, we have more of a demand for this sort of thing, and so there's more of a supply of it to fill it.
Well, that's why we're here at TWiV, to explain the virology. So, the newspapers may not get it right, but we'll try and explain it. And I'm glad to hear that we have hot news items that we can talk about. Yeah. It's not good because we don't want people to die. But
no, it's certainly unfortunate for the woman who died of bacterial pneumonia.
I think if I was a horse, I would have heard some good news in your first story.
Yes.
So,
don't forget to immunize your horse, Dick.
I won't.
All right, we have two websites we just want to point you to quickly. The first is called the Jenny McCarthy Body Count.
Right.
You know this, Dick?
I looked it up. I was shocked.
I think I emailed it to you guys last week.
I was shocked.
This is a website that someone has put up. I don't know who, but it is about Jenny McCarthy, the celebrity who speaks out against vaccines in a profane manner. So it shows the number of preventable illnesses and the number of preventable deaths. The numbers come from kids who haven't been immunized, and the suggestion is that it's her fault because she's very vocal, and a lot of people believe her.
Mm-hmm. Why? Because she took her clothes off for Playboy?
Because she's a celebrity, Dick. They don't listen to scientists.
Well, there are other celebrities out there telling you to immunize.
I know, but she's a very prominent. She was just in Time in April. She had a quote. You want me to read the quote for you, Dick?
Sure.
I do believe, sadly, it's going to take some diseases coming back to realize that we need to change and develop vaccines that are safe. If the vaccine companies are not listening to us, it's their effing fault that the diseases are coming back. They're making a product that's S. If you give us a safe vaccine, we'll use it. It shouldn't be polio versus autism.
Fortunately, it's not polio versus autism.
Yeah, I never heard anyone accuse polio vaccine of causing autism.
No.
So why use that one as an example?
Because she doesn't know any better.
Right.
I mean, folks, vaccines are great. They're really safe.
It's what keeps us here.
Anyway, the number of illnesses so far, 18,098. Number of preventable deaths, 160. I think this is a good website to publicize.
Yep.
Yeah.
Okay, and the last one is 10 dumbass reasons why people don't get their flu shots. Did you see this one, Dick?
I actually did not see that one.
So I won't read this, but we'll put a link in the show notes. It's by our friend Mark Crislip.
Uh-huh.
It's the sarcastic infectious disease physician from Canada. And he has 10 reasons, which is they're really funny.
Yes.
Here's number 1. The vaccine gives me the flu. And he writes, dumbass, it's a killed vaccine. It can't give you flu. Number 2, I never get the flu, so I don't need the vaccine. Irresponsible dumbass. So and on and on. It goes on and on. It's really good. Someone actually, who sent me the link? Everett. And I laughed a lot. So thanks, Everett. And now we're going to do some reader email. But before that, here's a message from our good friends at ASM, which is coming up in a few weeks, guys.
It is.
Live from ASM.
That should be fun.
It will be a lot of fun.
On May 17th through the 21st at the Pennsylvania Convention Center in Philadelphia, the American Society for Microbiology will hold its 109th General Meeting, the largest annual gathering of microbiologists in the world. Visit the General Meeting website at gm.asm.org to view the preliminary program, register for the meeting, or reserve your hotel stay. That's gm.asm.org.
Now we've got a bunch of reader emails. The first one is from Everett. Same Everett who sent us the 10 reasons not to get your flu vaccine. Some time back, I asked a question about pigs being better hosts for influenza, who then spread it on to humans. Naturally, I was incorrect. However, you brought up in the podcast that pigs serve as mixing vessels for influenza. Important parts, but not vital. So, the 2 kids in California diagnosed with swine flu leads me to ask, why don't we vaccinate pigs as well as humans? If they do serve as mixing vessels, wouldn't we have a better chance of staving off an epidemic if both the vessel, pigs, and accidental host, us, had immunity to a few strains? Any ideas, guys?
Expense.
Money.
Yeah, well,
that's probably the same. That was a tie, Alan. Yeah, farmers don't like vaccinating their animals if they don't have to.
But they do. There are other vaccines given to animals.
No, I know. Right? I don't say they don't vaccinate, but I think that they don't like to do it if it's going to cost them an arm and a leg.
Agriculture is a business with very, very slim profit margins, and so you have to have a vaccine where the benefit is going to outweigh the cost, and the incidence of swine flu is not enough to really justify that, probably.
And do the pigs die from influenza? Probably not.
They
if
don't
a pig
die,
looks like it's dying, you kill it.
So, in fact,
and then sell it.
A paper just came out in Journal of Virology where they infected pigs with the 1918 influenza strain, and they were fine. They don't get sick. Whereas in ferrets, it kills ferrets and mice.
See that?
So it's much less virulent in pigs.
Mm-hmm.
That would be hard to sell, something that doesn't cause a serious disease, right?
As a preventative for humans.
I don't know. Can you sell that, Dick? That's a tough one.
That would be very difficult.
But also, you'd have to immunize against a number of different strains because there are, besides swine flu, there are other avian and human subtypes in there. So you basically have to cover all of them. And
because the benefits of the vaccine don't accrue to the farmer, you run into this fundamental economic problem. You've got, this effect that somebody's got to buy the vaccine. The farmer buys the vaccine to benefit somebody who, is just out there somewhere. It might even be a vegetarian, so they're paying money for something that's not going to get them any direct benefit.
And now something completely different, although it's the same subject. Does anybody know how pigs acquire influenza, swine flu? Pig-to-pig transmission without humans. Do we know who Robert Shope was?
It's some kind of parasite that they eat.
Remember
I don't know if that's
Shope?
right. Is that correct, Dick?
Well, Robert Shope spent a lot of his life, Robert Shope Sr., at Rockefeller University, then Rockefeller Institute for Medical Research. He spent most of his adult life studying the transmission of virus infections and studied swine influenza from the standpoint of swine ascaris, called Ascaris sum. An Ascaris sum is a big long worm the size of a pen, but the eggs are quite small, and the virus is found in the worm as well as in the pig. So the eggs of the parasite are the stage of the parasite that lives in the environment and the pig ingests accidentally as they're feeding in the pig trough. The egg then hatches in the small intestine of the pig. The larva comes out, migrates through the tissues, and eventually ends up back in the small intestine to grow up into an adult worm. And at the same time, it then sheds the virus and gives the swine influenza to the next pig. So there's a vector for this, although it's a mechanical vector. It's not an insect vector. It's the pig Ascaris called Ascaris sum. He was widely respected for his views over the fact that if it wasn't for fecal contamination of the environment, swine influenza would be a lot less frequent.
I remember hearing about that when I was a student. I haven't heard about it since.
It's called a paratenic host.
So, that's from pig to pig.
Yep. Yeah, there are other things that do that, too. You can transmit Japanese bee encephalitis this way as well.
So it also goes from birds to pigs, and it goes from humans to pigs, and from pigs to humans.
That's probably through fecal contamination.
In fact, in this CDC article about the swine flu outbreak, they said up till now we haven't really had a very good screening program for pigs in the U.S., but now we're going to do it. We're going to check more and more pigs for flu. Makes sense.
Of course.
Okay, so that's your discussion on immunizing.
And of course, if they did have a vaccine for them, it would be called a poke in a pig.
Oh. Let's move on. Enio writes, thanks again for the show, Doctor. I hope you don't mind, but I took the liberty of posting a review in the iTunes store. 5 stars.
5?
Thank you, Enio.
Do we
in
mind?
That case, we don't mind.
Do we mind? Do we
thanks
mind?
Very much. We appreciate it. As for possible topics, is there any chance for segments of viruses for dummies? For example, I just listened to episode 8 with the person who had bone marrow transplanted and appears AIDS-free, but I don't understand how that would work. Viruses for dummies. Well, we try and explain things. So, since you asked, this is the person in Europe who had AIDS, and he received a bone marrow transplant, and he got bone marrow cells from a person who lacks the protein CCR5, which is a co-receptor for the virus. So, without this protein, cells can't be infected. Now, the bone marrow gives rise to lymphocytes, which are the cells that are infected by HIV. So, in this person, what they do is they get rid of his existing bone marrow by extreme irradiation, and then they give him a shot of bone marrow from someone else, and it grows, and it eventually populates his bone marrow and makes lymphocytes that don't have the receptor for the virus.
So it's kind of like an immune system transplant.
Exactly. Right. So that's it in a word. I hope that explains it. If not, tell us and we'll try again. But basically, he had a transplant with an immune system that's not susceptible to the virus. Susceptible meaning not having the receptor.
So, Vince, I will ask a dumb question too then, because then if you're missing that receptor, and let's say you're virus-free, what is the biological role of that receptor in normal human immunological biology?
That's a very good question. Apparently we don't need it, or as far as we can tell.
Maybe you're susceptible to something else that you haven't encountered yet.
I should say that the people, the person actually didn't lack the protein. The donor had a deletion of, I think, 32 amino acids.
Yeah.
And so the rest of the protein may still serve its function.
Or that protein protects us against some disease that we're not getting anymore.
Well, that's what I was...
Well, there's some thought that this protein is CCR5 might be a receptor for smallpox. And so the idea was there's quite a bit of the population that it has this mutation in CCR5 gene. And so the idea may have been that they may have been selected by smallpox for resistance to smallpox.
That means half of Europe has that deletion.
It's quite a bit. So that might be the remaining protein may be functional. To answer your question, there are no people that lack the protein entirely, so it could still work, but it doesn't work for a virus, which is good because that often doesn't happen. You take away what the virus needs,
and then you die.
Yeah.
That's right.
Jesper writes, I just posted a link on my blog to you. As I mentioned on the blog, I stumbled upon you when filling up my MP3 player for a week offline. I found you on sciencepodcasters.org. When I got back, I checked online bookshops to get Principles of Virology. Such a massive hole in my knowledge base could not go unfulfilled, only to find it to be way out of my budget. $200 in Sweden. Whoa. I'm sorry about that. When I heard about the link competition, I upped the priority of mentioning you in my blog. So he sent us a bunch of links to other science podcasts, because at the time we had asked for more, and we used actually some of those. Science Friday we used as a pick of the week before, and a couple of others. Brain Science podcast. And he writes, I just wish the day had more hours, so much to learn, so little time. Anyway, you guys are on my feed list now, and hopefully some of your knowledge will seep into my brain. Well, all the best. Keep up the good work. Thanks, Jesper. We appreciate it. Patrick writes, I'm a PhD student in Stuttgart, Germany. Although I'm now working on cancer, I was always interested in virus research and thus listened to your podcast.
Wait a minute. Some cancers are caused by virus.
Which ones, Dick?
Let's see. There's mammalian, Charlotte Friend. I used to know a lot of that stuff.
There are, as far as I know...
Mammary tumor virus.
Well, it's a mouse one. In people, HTLV1 and HPV.
Okay.
HPV,
right. Big ones.
And don't forget hepatitis. Right, you just said that, though.
Hepatitis B and C. Right. You're right, exactly. Now, my suggestion question is from Patrick. Since 2007 in Germany, there is an emerging disease in calves which pretty much looks like hemorrhagic fever to me. So far, nobody knows what it is or what it results from. Have you heard of something like this? So he sent us a link, and here, Dick, you might be interested in this. It's a link to a German newspaper, but it's in English. Holy Mary, help us in our hour of need. Germany's mystery cow disease. Mysterious illness is causing calves to bleed to death on German farms. Veterinarians are stumped over what is causing the deaths. Vaccines, genetically modified feed, or perhaps even the first mother's milk. So basically these are calves who are just take their first milk or they're vaccinated and they start bleeding and dying. And what can a cattle farmer do when he sees blood running from his calves like water when they become lethargic, febrile, and by the next morning are lying dead on the floor? What do you think this is? All right, so what I did is I put this up on Twitter. I don't know what it was. And some guy said, well, many years ago there was a disease in cows, I forgot where they were eating silage that had a mold in it that contained an anticoagulant. Do you know about this, Dick?
I don't, but I know that if they were doing rat control, they might have encountered the same thing.
What is the name of that?
I forget the name of it. We use it down here in the...
Coumadin.
Yeah, Coumadin. That's exactly right.
Yes.
Yeah, so this person said there was some incident where the silage actually grew something that had an anticoagulant. So that could be this, but I don't know. It does sound like a hemorrhagic fever, but it's very quick. And I don't think it would.
That sounds chemical to me.
Yeah.
It probably
it's
is
a toxin or a chemical or something.
Yeah, it does sound right.
So he says, thank you and keep up the good casting. Okay, thanks, Patrick. And this is from Twitter. I got the other day.
So it'll be short.
From Mendelt. It'll be less than 140 characters. Downloading episodes of This Week in Virology. Biotech is almost as interesting as computer science.
Wow.
Gee.
Almost, not quite. Well, anyway, we got a bunch of new listeners from Futures in Biotech, another podcast that I appeared on a few weeks ago.
Nice.
So we welcome you guys and gals, and we're glad you're joining us. And we're going to actually do an interesting Futures in a couple of weeks, so stay tuned for that. Swiss Compass writes, just wanted to bring your attention to the video below where transcripts for podcasts are discussed. So he has a video where you can have your podcast transcribed and have a written form. I think if there's any way you can incorporate this to your podcast, it would add a lot of value. for people who learn by reading while listening like myself, an enormous value for searchability by Google, etc., for archival reasons. That's a great one. It seems the transcripting service mentioned may not be that expensive, and even if it was a simple search for transcription services, returns a lot of results. What do you think? So I've looked into this because I think it's a good idea. In fact, Futures in Biotech is transcribed. There's a company in the UK that does it. And there are two options. One option that you pay for, we pay for it, TWiV pays for it, and then they post it for free for anyone, and it becomes Google searchable, and anyone can download the transcript. Or they do it for free, but it's kept on their servers, and people have to pay for it if they want to get it, and then it's not Google searchable. So I don't know how much it is. I have to look into that. But I think being Google searchable is best. That's my opinion.
Yeah.
That would be preferable.
Yep.
Yeah.
And as Alan says, we have such huge river of sponsorship money for TWiV that we could easily pay for ourselves, right?
We were just sitting around before the show pondering what to do with it. So
Exactly.
Cedric writes, "Hi, Vince, Dick, and Alan. I'm a biology undergrad and a big fan of the show. In addition to the witty banter and informative discussions, I really appreciate your podcast, book..."
Who's he listening to?
Yeah,
He's running the tape backwards, actually.
I really appreciate your podcast, book, and blog recommendations. I recently subscribed to Distillations per your recommendation. I wanted to share a few of my favorite science podcasts with iTunes and web links. So he mentioned Science Friday from NPR, which we've done. Science Talk from Scientific American. We haven't done that one.
No.
And The Skeptic's Guide to the Universe, which we have done. Keep up the good work. Thanks for the recommendations. We appreciate it. Hey, Alan, why don't you read the next one?
Well, okay. So Wayne writes in, thanks for reading my review of TWiV from iTunes. Well, are you kidding? We read all of our reviews obsessively. I've learned so much from your podcasts. I normally don't stray too far from astronomy, but I'm starting to get fascinated by basic virology and plan to read some of the books you've recommended. So we seem to be drawing people from all sorts of fields.
Look at that.
And Wayne goes on, he says, I'm very interested in how a virus type of life could evolve, or could viruses have devolved or become less complex? Is there any way to know? And this, of course, is one of the Zen koans of virology, I think. It's up there with Are Viruses Alive?
The problem is there's no way to know because there's no fossil record, right?
Exactly.
We don't have all the intermediates in the evolution. They go away, and that's a big problem.
But it's an excellent question. It is.
It's a great question. I mean, the endogenous retroviruses are probably the only fossil record we have. These retroviruses integrated into various genomes that have been there probably for millions of years.
Yep.
But we don't, and that's a problem. How did it evolve? We don't know. I mean, it could be that viruses were here in the beginning, and they evolved into cells. Some people think that, or vice versa.
And I think it's interesting that Wayne, as an astronomer, actually hit on the two primary ideas for how this could have occurred.
Right. So let me recommend to Wayne a wonderful treatise on this by Christian de Duve called Vital Dust, in which this wonderful molecular biologist that I had the privilege of working with at Rockefeller as part of his team, with Dr. Miklos Miller at the helm of the team that I was on, pondered the origins of everything, of course, like we all do, and sat down and actually put his thoughts into words and published this book called Vital Dust. And it's a wonderful book, easy to read, written for the general public, but with lots of details that you might be able to build on for your own intellectual growth and enjoyment. That's good.
I don't know how old this email is. It may be old, but we did a TWiV a while ago on evolution.
Yep.
With Luis. So Wayne, if you haven't listened to that, you should. There's some good thoughts in there, and there are a couple of books recommended there as well. Right. It often happens that people outside the field ask the best questions.
Absolutely. Yeah. Isn't that why you have me here? I don't always ask the best questions, but I certainly ask questions that nobody else in virology would ever think of asking.
Absolutely right. And that's why in a couple of weeks we're going to have Mark Pelletier of Futures in Biotech join us, because he'll ask good questions from the outside people. Sure. Mahmoud writes, Dear Dr. Vincent, I'm a pharmacy student in the German University in Cairo. Actually, I like your blog and I'm trying to catch up with your posts. I created a new blog about HCV to help awareness in Egypt because you know that Egypt has the highest prevalence of HCV infection. I'm inviting you to visit my blog. I appreciate it. We'll be so happy if you leave a comment there because I would like to develop it. Here's the link, and I'll put this in the show notes. Another thing, I know about your podcast and that you will choose a blog to be the winner. For that, I recommend viralmag.wordpress.com because it's interesting and simple enough for any readers to follow. Thank you for your time and waiting for your comment. I'm sorry, Mahmoud, I haven't left a comment, but I shall on your blog. And it's good that he's made a virus blog and that there's this other one. I haven't looked at it yet. I'll take a look. Are writes, "Hi, I just read an interesting article from the Telegraph about Creutzfeldt-Jakob disease, a.k.a. mad cow disease, and I realized with all the talk in the media about this disease, I still know very little about it. I'm not sure how well-informed citizens in the U.S. are when it comes to this disease, but there has been a major scare here in Europe because of all the deaths by it in the UK, but the newspapers haven't really covered any of the science behind it. So here's a suggestion. Could you have a short segment about this disease in one of your podcasts? I think many people would find it quite interesting, especially from this part of the world. I know it's not a virus or a bacteria, but you are the only source I could think of that covers these things in a sensible, easy-to-understand way."
Wow.
That's very nice.
It is.
With my sincere thanks for the effort you put into the podcast.
I'll be darned.
I think
should we do an episode of TWiP?
This week in prions. We will do a show on prions. I think it's a good idea.
Sure.
In the meanwhile, look at Virology blog, virology.ws. A few weeks ago, I had a post about this. But we'll do a show. Maybe get someone on who can talk about it.
Yeah.
But, it's interesting that you say that you want us to explain it to you, even though it's not a virus or a bacteria. The fellow who was instrumental in the prion hypothesis, Stanley Prusiner, came to the virologists to help him out early on because they're the only ones who would listen to him and his theory that this was a protein-based infectious agent. And he got the Nobel Prize for that eventually. But I think that's interesting that virologists understood him, the bacteria people and everyone, all the others.
Right.
This is nonsense.
Exactly.
Here's a little harder one. Vani writes, "Hey, Vincent and other TWivers. I was listening to the phage episode with a lot of interest. Great show as always. I was wondering about one of the discussions during the show. You mentioned that all phages were related, yet you also hypothesized that they are essentially parts of bacterial genomes that became autonomous. I was wondering how those two statements can coexist. I mean, if all phages are the result of independent genomic fragments becoming autonomous, how can they be related? A reason I ask is the following. A friend and former postdoc in the lab recently gave a very interesting talk at a meeting. He attempted to build a tree of life for phages. The result, as he called it, was a rake of life, not a tree. What this suggests is that there is not enough evidence for phages being related. Actually, he called phages the dandruff of life. His talk resulted in some very cool discussions between the two of us. I personally subscribe to the viruses may be the origin of life hypothesis more than a byproduct thereof. I was wondering whether you guys had some ideas or arguments that may fuel our discussion and maybe start discussions amongst other TWivers.
That's a very thoughtful email.
So, viruses are polyphyletic. They have many origins, and as we said before, it's hard to track them because you don't have any fossil record. We try to do it, but it's not often accurate. So I would point you to the previous podcast we did on viral evolution. There was also an article in But I also asked Max Gottesman to comment on this. So he responded. Max Gottesman, of course, did the phage.
On the show. That's right.
I am not sure I said that all phages are related, a hard proposition to prove, but certainly the phages that people are pulling out of our various places in the world show conservation. There is a vast collection of phage related to T7, for example, in terms of gene sequences and morphology. Temperate phage are closely related, sharing blocks of sequence identity. It is my impression that when a new phage is sequenced, most of the genes can be annotated based on knowledge of other previously sequenced phage. That phage can pick up bacterial genes and propagate them is a well-established fact. That this happened in history and contributed to phage evolution would be harder to prove. If, for example, phage picked up a bacterial DNA replication system, it would have to modify it beyond recognition. The needs of a phage are quite different from those of a bacterium. I mean, this is all the problem that we can't have a record and see how this works.
Again, allow me to interject just a moment. At one point in science's long history, they had a contest for the most meaningful sentence that characterized 100 years of biological investigation. And they had lots of interesting entries, and a lot of them revolved, of course, around Darwinism and Darwin's statements and this sort of thing, natural selection, etc. But the winner was it was a very interesting sentence that said, successful systems attract parasites. So that means parasitism is a multiply arising phenomenon. The moment there's a new system that succeeds during evolutionary selection, there are a set of parasites which arise to take advantage of that. And I think the phage are typical examples of this, where you wouldn't expect relatedness in disparate groups of bacterial species that have arisen, let's say, in the crust of the earth or at the bottom of the ocean or in a hot spring somewhere in Yellowstone Park. I bet you they all have phages associated with them, because that's how you get lateral gene transfer a lot, right? Sure. But these phages must have arisen independently of each other as well then, because those systems didn't exist before the phage was there. So it's a complicated circular, like, do proteins control DNA synthesis, or does DNA control protein synthesis?
Well,
the answer is yes.
I think the parasites also help the hosts as well, right?
Of course they do. I mean, they're not all bad, they're not all good.
I mean, the issue here is many of the phages that we're discovering in the oceans are actually unique. Most of the genes we don't know about.
Right.
The Max's statement that, they're all similar is not entirely correct.
Maybe the T phages.
Maybe the DNA replication enzymes, for example.
Yeah.
But there are many, many genes that aren't known. So we're limited by what we know so far. There's probably a lot more out there that we don't know about.
Yep.
Josh writes, hey, Dick and Vincent, I'm a second-year medical student at the University of Dundee in Scotland.
Yep.
I have exams coming up next week and have found your podcast to be a great way to revise and relax at the same time. Although I'm very early in my training, I already have a keen interest in infectious diseases, both viral, parasitic, just for Dick, and others. I found your podcast last week through Futures in Biotech and have so far listened to almost all your past episodes. I think they are based at just the right level of scientific knowledge, whilst also being very educational. I think a future episode about antiviral drugs, mechanism of action, or possible methods of future rational design would be particularly interesting, as these topics are not covered in particular detail at medical school. Thanks for the great informative podcast.
In fact, what is covered in detail in medical school? I mean, I've been teaching here for 38 years, Vince. How long have you taught here?
27 years.
Do we have time to teach in detail?
No,
I claim that what we do is give them an executive summary of an executive summary. No, it's really too bad that we can't go into details, and I appreciate that comment very much, by the way.
But then medical school will be 14 years long.
Well, the point is that this guy found some time to learn this anyway, so there must be some time to add some details to this.
Well, I think podcasts are a good way to, as he said, you can,
totally.
At night you're relaxing, you just listen to one.
That's a great idea,
I mean, a half hour, you can absorb some information.
So why don't we incorporate podcasts into the curriculum?
Should be.
Vince, sure. I think that's what we want to do.
Well, they don't certainly have TWiV in their curriculum. But I think the people who teach here don't even know about TWiV.
But guess what? Here's a way to get in there even when they don't want us.
Good idea. Zach writes,
I feel like a prophage.
You are a prophage. Hi, I just wanted to let you know I've become a bit of a fan of your show. I have been collecting science podcasts to listen to improve my science knowledge. Mostly I've been picking up astronomy podcasts, but my father pointed out yours. I very much enjoy it, but parts of it are over my head. I hope with enough listening I will understand more. By the way, his father is on the faculty here, and we shall have him on TWiV in a few weeks.
Here's another great segue then. Mm-hmm. Who was Frank Hawking's, Stephen Hawking's father? Oh. What was Frank Hawking's livelihood?
Astronomy?
He was a nematologist.
Hey, there you go.
He studied worms. I actually knew him.
You knew him, yeah?
Yeah.
Should we get him on the show?
I don't think he's alive anymore.
If you have not checked it out yet for your science podcast shout-out, check out 365 Days of Astronomy from the IYA New Media Working Group. And then he lists a few other science podcasts, some of which we've mentioned and others which we'll check out. Thanks a lot, Zach, and stay tuned for your dad. Jessica writes, hello all. Thank you so much for your fantastic podcast. Recently you mentioned that influenza transmission was higher in seasons of low humidity, since the virus can stay in the air longer in the absence of excessive humidity. However, if this is true, why does influenza transmission appear to occur year-round in particularly humid areas such as Southeast Asia? Additionally, while I've heard it said many times that influenza occurs year-round in Southeast Asia, I'm wondering if there is any serological data to support this notion, or if it is merely based on reports of influenza-like illnesses in the region. There's definitely serology to support it, so it is flu. Understandably, Southeast Asia does have a mixing pot of humans, poultry, and swine living in close proximity, but does that alone account for the continued transmission? What other factors aside from humidity account for the seasonality of flu in most regions? Are certain strains such as A more likely to persist year-round because of their ability to infect birds? Yes, Dick.
I have a thought.
Sure.
Maybe it relates to the migratory routes of wild birds and the timing.
Peter Palese said no. It's the virus moves around from south to north hemisphere by humans, basically, travel. There is some component of the birds, but humans play a
Wild birds
role.
Don't play any role?
They play some role, yeah.
Some.
Yeah, well, I don't know how much.
Maybe the initiation of the outbreak.
Well, here we're talking about areas where it's endemic all year round.
I mean, getting a new virus strain going, maybe that's what I meant to say.
That's a different story, yeah.
Because, I mean, we've already seen the old ones, we've responded to them, and here's the new one that pops up. And when? Just after the migratory route of the birds are finished.
Well, they originate in Southeast Asia.
Yep.
The idea being that humans and animals are close, and you get this interspecies transmission, right? But she wants to know, is it that or is it something else?
Well, those are two different things, though. Transmission of person to person and transmission from animals to people. Those are two different ideas.
Well, all right, so let me finish her email then. Well, could the flu virus mutate in such a way to allow it to sustain transmission no matter what the season? Again, I really enjoy your podcast and would appreciate any insight you could provide into this topic. So the transmission, why is it year-round in the tropics? We don't know. Peter Palese, I asked him that on Futures in Biotech. It's an unknown. One possibility is that in these highly humid, hot tropical climates, it's more of it is a contact transmission rather than aerosol. Okay, because under those aerosol, humidity, and heat conditions, it shouldn't work. And that's what controls seasonality in temperate climates. So it must be another mechanism of transmission. Could the virus mutate? It could, but there's no evidence for that. There's zero evidence.
Well, even if it did, though, what's the nature of the mutation that would take advantage of a humid environment?
Well, it would have to change the structure of the virus significantly, I would think. Not likely. Are certain strains likely to persist year-round? Because the birds have every known human strain in them, subtype, as well as all the bird subtypes. So they're the reservoir. Birds are where everything is. So the answer is yes, they persist forever in birds, and that's the origin of new strains. But the annual seasonal flu, it was here in the winter. Now it's going to be going in the southern hemisphere, which is beginning their winter, right? And then it will come back. And a large component of that is human travel, but it's also the avian transmission of the virus. But, the main question you have, we don't know the answer. I'm sorry. I don't know why it's transmitted year-round in human areas. But again, the idea is that it's contact and not aerosol. Derek writes, keep up the good work, gentlemen. You're doing a great job. I really look forward to listening to your past podcasts over the summer. That's a good idea.
Sure.
Listen to it over the summer.
Reruns.
Lucas writes, hi, Vincent, Dick, and Alan. My name is Lucas. I'm writing to you from sunny Melbourne, Australia. Right now, I'm an enthusiastic undergraduate student at Monash University studying molecular biology and genetics. In the absence of some unforeseen disaster, I will be starting postgraduate studies in plant genetics from the middle of the year. I've never written to the presenters of a podcast before, but I thought it was important to make sure you understand what an excellent thing you are doing. I can say to you all with a serious expression on my face that This Week in Virology has become one of the major pleasures in my life. I've listened to every episode and generally listened to every episode a number of times. I find it a wonderful way to learn more basic science. Also appreciated are the observations that you and your guests make about the process of scientific research.
Wow.
I hope you're aware that it's really valuable stuff that you've been doing, and that there are lots of us out here who appreciate it. Thanks for all the time and effort. I tell everyone I know to listen to your show. I hope everything is going well for you guys. Thanks, Lucas. That was very nice. We do appreciate hearing
nice. Absolutely.
If you guys out there like what we do.
Hey.
And we get a lot of feedback. We appreciate it. But we think there's a need for this, and we love this stuff. So
we also enjoy it.
We enjoy it. I love it. All right, we'll do a couple more and then wrap it up. Jim from Virginia writes, on Futures in Biotechnology, Dr. Palese said a virus the size of your fist is comparable to a cell half the size of the Empire State Building. In this analogy, would a bacteria be about the size of a car? Yeah. That would be about right.
Mm-hmm.
Yep. I'm a layman, but appreciate excellence when I hear it. Thank you for doing such a great job. Please consider a program where you speculate on the impact of global warming on virology. Oh, lastly, is there any fossil record for viruses? Dick, global warming?
Yeah, well...
Thank you. Thank you for your answer.
Come on, I can expand on this if you'd like, but I'll leave it at that.
Give us an example of,
sure.
Of an impact.
I mean, well, you have to look at the way viruses are transmitted, and I think if you look at the arboviruses, then there is a direct connection between global warming, the expansion of not only the range of each of these vectors, be they mosquitoes or sandflies or things like this, but their growing season is extended also. So you can have more of them more often in more places. And for that reason alone, common logic suggests that global warming will have a negative impact on our lives because it will expand the life of those vector species, which can then transmit it for a longer period of time. I don't think global warming per se is going to have an effect directly on the agents because they're inside of us. And global warming is not something that affects us per se because of our homeostatic mechanisms for regulating our own biological rhythms. It relates to the way they're transmitted versus the way they behave once they get inside of us.
I mean, it's definitely something where you see the vectors expanding their ranges, but at the same time, you have to kind of temper that with the realization that as you get further north, often you're also going up into more developed countries, just because that's the way history is played out. And as you go, say, from Latin America into North America, you actually see there are a lot of barriers to transmitting some of the arboviruses, like more widespread use of air conditioning. So people are getting bitten by fewer mosquitoes. So you may not see, it won't necessarily be the case that we'll see malaria rates in New York reaching the same level as they are in Haiti. But it's still, obviously, you worry when Anopheline mosquitoes get a wider range, for example.
Sure. I was thinking of the countries that they're already in, Alan. Like, for instance, in Kenya. Kenya has this wonderful city, Nairobi, which is it's a very high altitude city.
Right.
It's over a mile high. And because of that, the mosquitoes hadn't found their way up to that level because of the climate. Okay,
right.
But now they're there.
Okay.
They're in the heart of Nairobi. Okay.
Right.
Oh, I wanted to go back and just recommend a book to this gentleman who has difficulties imagining how big things are relative to each other.
Mm-hmm.
There's this fabulous book called Powers of 10.
Oh, yes.
And there's also a video version of this that teaches you how big things are.
The author is who? Do you know?
It's by Scientific American. I think they actually took it over. I have the book, and I have the DVD also. It's an absolutely fabulous treatise.
All right, I'll look it up and put a link in.
You'll love it. You'll just love the book.
And as far as fossil record, we talked about that just a little bit, right?
Yeah.
There isn't any except for the endogenous integrated retroviruses, which help a little, but not entirely.
What if the woolly mammoths died out because of a viral infection, but they're frozen, and we're now unfreezing them?
And the virus will come out? Well, it could be, but a lot of these endogenous...
That's not a fossil, obviously. It's not a fossil.
I think you should option that to Hollywood.
I think it would be really... Well, the mammoth genome was just sequenced, right?
It was.
And
come on, that's how they got the 1918 flu virus.
Well, that's by pulling out from pathology.
From frozen...
Right. No, that's totally plausible for a reasonably small virus in a well-preserved sample. I mean, these are army samples, so they're taken care of. But woolly mammoths are not as well preserved.
Do they have any viral sequences from amber?
Not that I know of, but the mosquitoes in the amber, yeah.
No, they have other sequences, though, they got from those animals.
Not that I know of, but that would be interesting. I want to know if the mammoth sequence has retroviruses in it. I assume it does. I just haven't looked at it. I have to do that.
Yep. Or Neanderthal.
Sure, absolutely. Okay, let's do one more email. Ben. Now, Ben sent us 1, 2, 3, 4 emails, so I'll read the first one. But we really appreciate this, Ben, because they're fabulous. They're all fabulous. First of all, I want to thank you both very much for a thoroughly enjoyable and educational podcast. I'm very glad I discovered your podcast last week via a reference in This Week in Biotech, or Futures in Biotech, I think, and I am working my way through your episodes. I am almost caught up. I am indebted to both of you, as well as the University of Columbia, for making this possible. I'm writing to make 2 points and to offer some information. A, in the episode Following the video game episode, someone wrote a question about whether epigenetics could result in inheritable traits. The concept of epimutations was dismissed, but here is an example. So he sent a reference to a paper. This is just beautiful. Inheritance of a cancer-associated germline epimutation. So it turns out there are methylations of DNA sequences that are inherited. So the gene sequence is the same, but the methylation pattern, which affects expression, is inherited. So in this paper, persons who have a methylation of one allele of a gene called MLH1 in somatic cells throughout the body, this is called a germline epimutation. Right? It's not in the germline. Have a predisposition for the development of cancer in a pattern typical of hereditary colorectal cancer. So in this paper, they studied the inheritance, and they show that this epimutation, is transmitted from mother to son, but erased in his sperm. So it is somatic mutation. It's methylation. It's not a gene change, not a sequence change, but it's methylation. So remember a long time ago, Dick, I complained when people say genetic mutations?
Yeah.
And then we said, well, there's no other. And someone said an epigenetic. And I said, yeah, but it's not genetic. It's not inherited. And this is what he's talking about. They can be. So this is a methylation that is inherited. We don't understand why. Isn't that interesting?
Yes.
That's really cool.
Thanks for that. That's great. And now, well, this one is tough, more philosophical. We all anthropomorphize evolution, endowing it with cunning, goal-directed behavior. It is also common for us to periodically lament this habit as not just sloppy thinking, but also as false. You did this on a recent episode. I'm writing to defend the concept of intention as an accurate abstraction. In fact, no less accurate than when applied to a human mind. As with all abstractions, it glosses over details. As with all abstractions, it is in some contexts sufficient, while in other contexts, the details glossed over by the abstraction turn out to be important. It is no coincidence that algorithms based on evolution are routinely used in the field of artificial intelligence, as I assert that evolution is a form of intelligence. As it happens, it is a form of intelligence which eventually invented other forms, such as endocrinological learning systems, and eventually neural networks to do the things that it was too slow at, which in turn invented computers to do the things they were too slow at, etc. Granted, we aren't very good at making computers be goal-directed, but that is already changing. The day may come when we can explain and predict human behavior via the language of volts and grams, but when that day comes, it will not render obsolete the concept of intention as applied to human thought. I apologize if this is a bit off topic, but this common lament is a pet peeve of mine, as I believe it sells short the clever engineering work performed daily by evolution, and also causes needless confusion in the field of artificial intelligence. Why would the biotech industry be interested in evolving enzymes that perform specific desirable functions, if not because evolution is in some context still a more able engineer than the human brain? If you disagree, I challenge you to devise an empirical definition of intention which applies to the human brain but not to evolution. What do you think, guys?
Well, who wants to go first? Because I do have an opinion about this. That it's almost an agreement with this gentleman's. And that is if you look backwards, you can't look forward in evolution, but you can certainly look backwards. And if you look backwards as far back as you want, as far back as we can, to the speculation of how life actually arose on this planet, assuming that it arose on the planet and it wasn't brought in by some comet from some other place that it already existed, you have to be impressed with the fact that it is directional in the sense that it advances from simple to complex in every respect. Simple systems first, and the simple systems then become somehow more complex and more redundant to have backups to make sure that they don't fail. So that it's almost like building an airplane. The first airplanes didn't have any backup whatsoever, no fuses, nothing like this, and they crashed a lot. But a few of them survived, and you can learn from the crashes to invent more backup systems so that things don't go haywire again. So if you look at the way a cell behaves, and then multiple cells behave, and then organisms, and then finally us, I have a philosophical definition of evolution, and that is the purpose of life is to understand itself. So eventually...
It's an intent, right?
We will end up with a life form on this planet that understands how it got there. And isn't that what we want to do? We want to understand everything. That's why we're in business to begin with.
Yeah, it's true. I can't argue against that.
So I don't know how far we can take this forward, but I can certainly say that an amoeba can't do as fast a thinking process as we can. They didn't have any TWiVs, by the way. They didn't have any broadcasts like that. None of them went to the moon, although we took a lot with us when we did go. So it sounds heretical to say this because obviously there shouldn't be a direction to evolution. It should be a random walk, as everybody thinks of it. But it's not random in the sense that when something arises that is superior to something that was there before, that the superior thing is kept. And in fact, a lot of the other stuff is kept too, as a backup. I'm always impressed by the other uses for proteins, like the lens protein in your eye used to be a serine protease. How does that work? We don't throw anything away, we just reuse it for something else. And that, for me, is directional.
Yeah, I think this comes down to exactly what you started with, Dick, which is that you can't say where evolution is going to go. You can only look back on where it's been and show where it got to. And you can make some predictions about, if we apply this selective pressure, we'll get something that has adapted to it.
Right.
But even when you're evolving industrial enzymes, as Ben's example is, you can't say, well, we're going to evolve an enzyme that has this specific base sequence or this specific amino acid sequence. You say we're going to evolve an enzyme that breaks down this chemical compound.
Sure.
And you don't know exactly how it's going to do that, but you do know that if you apply this pressure, you will get this result.
That's right.
So I think the issue is there is intent. If you look back on this and you see the sequence of events that led up to the present, you would have to say that the present situation was the result and therefore, in some sense, the intent of this whole sequence of events. But that doesn't mean that this was the inevitable outcome. There were billions of other possible outcomes that could have occurred that would have been equally intentional.
Right, but I think the rhetoric is that all these arise at once, and then the environment changes, and all of a sudden the one that arose over here was the superior one for that new environment, not for the old one, but for the new environment. And that leads you to a whole new series of random mutations that could be in wait for the next environmental change.
Right.
So I think global warming, if you want to know what that really means, that's really fast-forwarding the evolutionary process.
I understand that. It's a good point. And I think I was probably narrowly interpreting intent. Ben tells us a bit about himself. He's in the IT industry. And he says, I don't understand how anyone living in this age could bear to miss out on the excitement of discovery spearheaded by academic researchers such as yourselves. And he said he's gone on to look at podcasts and various journals to learn about this. And now he's bought a Kindle, which is one of these book reading. And he thinks that now his consumption of written word is more balanced with podcasts. Yours is the latest addition to the information sources I follow. Thanks for sharing the excitement of discovery with the rest of us. But, a written word is very different from what we're doing, even though it's more portable as a result of the Kindle. The spontaneity that we have, the conversation. This is a conversation. You can't do that on a Kindle. So there will always be a role for TWiV.
Yep.
All right. We have a couple of picks of the week. Science blog of the week is called Science-Based Medicine, exploring issues and controversies in the relationship between science and medicine. The editorial staff is composed of several physicians who, alarmed at the manner in which unscientific and pseudoscientific healthcare ideas have increasingly infiltrated academic medicine and medicine at large, have decided to do their part to examine these claims in light of science and skepticism. So it's a good blog, and our friend Mark Crislip writes for this, as well as Dr. Novella, who is the Skeptics podcast that we talked about.
Okay.
He's involved in that. So it's very good. Science podcast of the week is WNYC's Radio Lab.
Right.
Which I've been listening to because of you, Dick, and I like it a lot. Science meets culture and information, sounds like music, a patchwork of people, sounds, stories, and experiences centered on one big idea. You'll be in it in the fall, right?
That's true.
We look forward to it. It's really crazy. There's all stuff going on all over the place. They must do so much editing for that.
They do.
And Science Book of the Week is Flu by Gina Colada.
Gina Colada.
Story of the 1918 pandemic. The Great Influenza was a pick previously by John Barry, and this is another book, different, good, and it's really nice. I like it. So take a look at sciencepodcasters.org, promednetwork.com for other science podcasts. And a general meeting is coming up, ASM, live TWiV, the ASM general meeting, May 19th. I think we'll have a room for you pretty soon, or a link to watch the streaming video. Thank you for writing reviews on iTunes. We're now on the front page, the featured page on podcasts, Science and Medicine/Medicine. Thanks for everybody for doing that. Keep it up if you'd like. We are number 10 today on the most subscribed podcasts, so that's great. We appreciate all your support. We can't do it without you, so thanks very much. But do keep sending us your questions and comments.
Absolutely.
TWiV@TWiV.tv, and we will routinely do a massive Q&A such as this one.
And for those of you who didn't hear your letter yet, be aware that we have several more in the queue already that we're looking at.
Absolutely.
Yeah, I didn't get to all of them today, and
we haven't lost you.
We haven't lost you. We will read every one because we really appreciate it. But that's what this podcast is all about: interaction with our listeners.
Hear, hear.
You know, we want to know what you want to hear about, and so we'll do that. Gentlemen, thank you very much. I enjoyed that.
It's a pleasure, as usual.
Was it good to do Q&A?
Oh, yeah.
Yeah.
Okay, you've been listening to This Week in Virology, the podcast about all sorts and kinds and permutations of viruses. Thanks for joining us. We'll be back next week. Another TWiV is viral.